Imagine a tumor, like specifically a BT474 breast cancer xenograft, right? Which is basically a human tumor growing in a controlled lab model. Now, this specific tumor has completely stopped responding to one of our absolute best cancer drugs, T-DXd.
Right, which is highly unusual.
Yeah, I mean, it's a drug that usually just decimates this type of cancer. But right now, the tumor is growing and the drug is, well, just bouncing off. The clinical window for you, the listener, if you were the patient, is closing fast. But then, and this is where it gets crazy, scientists do something totally wild. They inject two separate, completely harmless-looking molecular puzzle pieces into the bloodstream.
Just two tiny fragments.
Exactly. Those pieces find the tumor, they physically click together inside the living body, and suddenly the drug works again. The tumor just shrinks.
It completely reverses the drug resistance. I mean, we are effectively using the patient's own circulation to assemble biological missiles right on top of the target.
It's mind-blowing. Welcome to today's deep dive. We are jumping straight into a really dense, highly technical July 2026 Nature paper. It's titled Modular in Vivo Antibody-ADC Click to Reverse Drug Resistance in Tumors.
Quite a mouthful, I know.
Yeah, totally. But our mission today is to exhaustively decode every mechanism, every term, every little detail of this paper for you. We're translating some seriously complex biochemistry to understand exactly how scientists are, turning the human body into a chemistry flask to outsmart cancer.
Which is such an exciting frontier.
It really is. So before we can understand this click solution, I think we have to look at the weapon that's currently failing in that BT474 tumor. I'm talking about ADCs, right? Antibody-drug conjugates.
Right, yeah. So ADCs, they're these engineered molecules, and they have three distinct parts. You have the antibody first, which acts as the homing device.
Right.
It's designed to find and bind to a specific antigen, like a flag, on the surface of a cancer cell. Second, you have the cytotoxic payload, so that's the actual toxic chemotherapy meant to kill the cell.
The warhead, essentially.
Exactly. And third, you have the chemical linker that connects the homing device to the payload. When we look at FDA-approved examples like T-DXd, which is trastuzumab deruxtecan, or TDM1, they are specifically designed to hunt down the HER2 receptor.
Right, and HER2 is heavily overexpressed in a lot of breast and gastric cancers, right?
Yeah, precisely.
So the logic for you listening is pretty straightforward. Find the HER2 receptor, drop the payload, kill the cell. Like a mail carrier who can only deliver packages to houses with a very specific bright red mailbox. The HER2 receptor is the red mailbox.
That's a great way to put it. But for an ADC to work, every single cell in the tumor needs to have a high density of those red mailboxes. And cancers, well, they are chaotic. This paper maps out a concept called tumor heterogeneity.
Meaning the tumors are just one uniform block of identical cells.
Exactly. The researchers didn't just look at HER2, they also looked at a completely different receptor called EGFR. And using this automated tissue imaging, they mathematically mapped where these two different receptors actually live within the exact same tumor.
And what did they find?
Well, they use a mathematical metric called the dissimilarity coefficient, or DSC. It basically measures spatial overlap. So a score of 1 means the HER2 and EGFR receptors are sitting on the exact same cells in the exact same spots.
Okay, a perfect overlap.
Right. And a score of 0 means they never touch. The imaging revealed a DSC of 0.12.
Wait, 0.12, I have to push back on the biological logic here. If the DSC is 0.12, these receptors aren't even in the same neighborhood.
No, they're really not.
I mean, you have cells with high HER2 and almost zero EGFR and vice versa. If they are completely isolated, sending a highly targeted HER2 drug just feels biologically doomed for the EGFR-heavy regions. Like, why would researchers even try to target a tumor that is actively hiding its primary targets?
Because it gets even more insidious than just hiding. When these two receptors do happen to be near each other in that tiny 12% overlap, the EGFR can actually force the HER2 receptor into what we call a heterodimer.
It's heterodimer.
Yeah, it basically pairs up with it and physically alters its behavior. This heterodimer physically prevents the cell from internalizing the HER2 targeted drug. So not only is the target missing in 88% of the tumor space, but where it does exist, the EGFR essentially barricades the door.
Oh, wow. Okay, so if we just pause for our first recap here, making sure we have this mapped out for you guys. Bullet 1, traditional targeted therapies like ADCs often fail because tumors are spatially patchy. The targets just don't overlap, which this paper proved with that incredibly low DSC score of 0.12.
Right.
And bullet 2, the cancer cells actively alter their receptors to lock the drugs out using those heterodimers. So the question I want to throw to you, the listener, is this. If a tumor is actively hiding its targets, its mailboxes, and barricading its doors, how would you force the delivery of the package?
Well, the answer is you don't send the entire package all at once. You break the delivery into two parts, which brings us to the click solution.
Yes, the puzzle pieces. Let's get into it.
Right, so the scientists took an FDA-approved antibody that targets EGFR, a drug called pinitumumab, and they attached a tiny chemical tag to it called a TCO moiety.
TCO, got it.
Which stands for transcycloctene. Then they took their HER2 ADC, the trastuzumab ADC we talked about earlier, and attached a different tag to it called the tetrazine, or T's moiety.
Okay, so I'm picturing TCO and tetrazine like two halves of biological Velcro, or two interlocking puzzle pieces.
Yeah, that visual works perfectly for the physical mechanics of it, but chemically they operate under a principle called bioorthogonal chemistry.
Bioorthogonal. That's a heavy word.
It is, yeah. Basically, the word bioorthogonal means they intersect biology at right angles, meaning they are entirely non-interacting with native biological systems.
Wait, so they just float around and ignore everything?
Exactly. These two chemical tags completely ignore everything in the human body. They don't react with blood plasma, healthy tissue, enzymes, normal proteins, nothing. They drift through the bloodstream completely invisible to human biology.
But they aren't invisible to each other.
Right. No, definitely not. When the TCO and tetrazine finally meet, they trigger a massive reaction. The paper calls it an inverse electron demand Diels-Alder cycloaddition.
Say that 3 times fast. Inverse electron demand Diels-Alder cycloaddition.
Right.
Okay. What does that actually mean for the molecules?
Well, the tetrazine is electron poor, and the TCO is extremely strained and electron rich. So when they collide, they aggressively snap together, releasing nitrogen gas.
Oh, interesting.
Yeah. The release of the gas makes the reaction permanent and irreversible. They form a solid covalent bond. And what makes this viable for a living organism rather than just working in a petri dish is the reaction kinetics. The paper notes the K2 rate is greater than 100,000 per molar per second.
Which in biochemistry means what exactly?
It means it is essentially diffusion limited. The absolute millisecond they bump into each other in the bloodstream, they clip together permanently, instantly.
Okay, let's walk through the actual sequencing in the living model for the listener. How are they deploying this two-part system in reality?
First, you inject the EGFR antibody, the one carrying the TCO tag. You let it circulate in the blood, it finds the tumor, binds to those EGFR receptors, and just parks there. And then you wait 24 hours.
24 hours? Why so long?
It gives the body time to clear out any excess antibody that didn't bind to the tumor. You want a clean slate in the bloodstream.
Gotcha. So, 24 hours later, the tumor is essentially peppered with these parked TCO tags. Half of the Velcro is pre-positioned perfectly all over the cancer cells.
Exactly. Then you execute step 2. You inject the HER2 ADC, which carries the toxic payload, and the tetrazine tag. As this second drug flows past the tumor in the bloodstream, the tetrazine instantly clicks onto the TCO tags that are already parked on the EGFR receptors.
So you've just assembled a multi-part therapeutic complex right on the surface of the tumor, inside the body.
Yes. The body is the chemistry flask.
Let's lock this concept in for a moment with our second recap. Bullet 1. Click chemistry allows two bioorthogonal molecules, TCO and tetrazine, to instantly bond inside the living body without interfering with any normal biology. Bullet 2. They sequence it by sending the homing beacon first, waiting a day, and then sending the warhead to snap onto it.
Yeah.
So audience, here's my question for you. Can you see the logistical brilliance here? Sending a drug in 2 pieces and letting the tumor itself act as the assembly line.
It really bypasses so many traditional delivery problems.
It does. But, you know, the chemistry working perfectly in blood plasma is one thing. How did they prove this molecular Velcro is actually getting inside the cancer cell in a living body? Because clicking on the outside isn't enough, right?
No, that is the crucial next step. They had to prove internalization. Like you said, it's not enough to build a drug on the outside of the cell. The toxic payload has to be swallowed into the cell's digestion centers, which are known as endolysosomes, to actually kill the cancer.
Right, the drug has to get in to do damage.
To track this, the researchers use something called a Frodo dye.
Oh, the Frodo dye. I love this part. This dye is fascinating because it acts exactly like a glow stick. You know, a glow stick doesn't emit any light until you physically crack it.
Right, yeah.
Similarly, this Frodo dye only fluoresces or lights up when it enters a highly acidic environment. And the inside of a cell's endolysosome is highly acidic.
Precisely. It's a perfect visual indicator. So they tagged the Click ADC with this Frodo dye. They also stained the tumor cells for a specific protein called MMP1.
MMP1, what does that do?
LMP1 is a biomarker that exclusively highlights those acidic endolysosome vesicles, so it's basically a map of the cell's stomachs. When they observed the cells under a confocal microscope, the glowing red dye perfectly overlapped with the green LMP1 markers.
Oh, wow.
Yeah. This proved visually that the cell was literally eating the assembled drug. Internalization was happening.
So, seeing it under a microscope is still an isolated environment, right? The real mic drop moment in this paper is the in vivo imaging, specifically the bilateral mouse model. Let's break down this experiment because the design is just brilliant.
It is a beautifully controlled setup. So a xenograft, as we mentioned at the start, is human tumor tissue transplanted into a mouse. In this bilateral model, they place two completely different tumors into the exact same mouse.
Okay, so a tumor on each side.
Yes. On the right shoulder, they grafted an NCIN87 tumor. Now, this tumor is HER2 positive, but EGFR low. On the left shoulder, they grafted an A431 tumor, which is the exact opposite. It's HER2 ultra-low, but EGFR high.
So on the right shoulder, you have an abundance of the traditional HER2 targets. On the left shoulder, you have almost no HER2 targets, but plenty of EGFR targets.
Right. And to see where the drugs were going, they attached a radioactive isotope, copper-64, into the drugs and put the mice in a PTT scanner.
The glowing radioactive tracker.
Exactly. When they injected just the standard HER2 ADC without any click tags, just the normal drug, the PT scan showed it going straight to the right shoulder, the HER2 positive tumor. It basically ignored the left shoulder entirely.
Which totally makes sense. If the tumor doesn't have the target, the drug just floats on by.
Yeah. But then they tested the click strategy. They pre-targeted the mouse with the EGFR antibody tagged with TCO, waited 24 hours. Then they sent in the HER2 ADC tagged with tetrazine and the copper-64 isotope.
And the PET scan lit up the left shoulder.
In a massive way. In the HER2 ultra-low tumor on the left shoulder, the exact tumor where the standard ADC completely failed the click strategy increased drug uptake by 3.2-fold.
I have to stop and trace the biology here because this is wild. If the left shoulder tumor has almost zero HER2 receptors, how does a HER2 targeted drug physically get inside the cell? The front door is locked.
It's a molecular Trojan horse.
A Trojan horse.
Yeah. The HER2 drug isn't using its own biological key to get in. Because of the rapid click chemistry, it flows by, clicks onto the EGFR antibody that is already parked on the surface, and essentially piggybacks its way inside.
Oh, I see.
The cell naturally internalizes the EGFR receptor to recycle it, right? And because the HER2 payload is chemically welded to it via the click tags, it gets dragged inside right along with it.
Yeah.
The cell thinks it's just pulling in its normal EGFR, but it pulls in the toxic payload too.
That is sneaky. Okay, time for our third recap for the listeners. Bullet 1. Using acid-sensitive Frodo dyes and copper-64 PET imaging, scientists proved that by clicking drugs together, a drug meant for one target can hitch a ride into a cell using a completely different receptor. Bullet 2. This Trojan horse mechanism bypassed a locked receptor by piggybacking on an open one, increasing uptake by 3.2-fold. So, audience, think about this. If we can Trojan horse a cell into eating a drug it normally rejects, what other undruggable diseases or highly resistant cancers could we sneak treatments into?
Which brings us directly back to the ultimate test, real-world efficacy and safety. We started this deep dive with that BT474 xenograft clinical case, the tumor that was completely resistant to our best drug, TDXD.
All right, the drug was just bouncing off because of those heterodimers locking the door.
Yeah.
How did the click strategy actually fare there?
In the study, they treated these resistant mice with the click therapy. They used pertuzumab, an antibody that binds to a different part of HER2, and tagged it with TCO. Then they sent in the TDXD tagged with tetrazine, and the result was a total reversal of the drug resistance.
Wow.
The Trojan horse delivered the payload despite the tumor's defensive heterodimers. The tumor shrank significantly.
But here there was a major engineering hurdle the researchers had to solve before this could be considered safe for humans, right? When they first tried this, the manufacturing process actually caused severe toxicity.
Yes, and this is a critical lesson in pharmacology. It's not just about what the drug does, it's about exactly how it's built. Initially, to attach the TCO and tetrazine tags to the antibodies, they used a standard chemical method called an NHS ester reaction.
NHS ester reaction, what does that do?
This method targets primary amines, specifically amino acids called lysines. A typical antibody has about 80 to 90 lysines scattered all over its 3D structure.
So using the NHS ester reaction means the tags attach completely randomly, right? You might have an antibody with 3 tags on one side, another with 12 clustered around the binding region. It's totally uncontrolled.
Exactly. It's highly heterogeneous. And the biological consequence of that random clumping is severe. The body's filtration system, specifically macrophages in the liver, recognizes these highly modified, randomly clustered antibodies as damaged proteins, or basically garbage.
Oh no, so the liver attacks them.
Yes, the liver aggressively pulls them out of the bloodstream. This rapid accumulation caused significant liver toxicity, which we measure by tracking liver enzymes in the blood, specifically ALT and AST. In the early tests with random conjugation, those ALT and AST enzymes spiked dangerously high.
So you shrink the tumor, but you destroy the patient's liver in the process. It's like trying to attach Velcro to a car by just throwing super glue and Velcro strips randomly at it. It works, but sometimes you glue the doors shut and ruin the paint.
That's a very apt analogy.
So how did they upgrade the engineering to actually fix this?
They pivoted to a site-specific conjugation method. Instead of relying on random lysine attachment all over the molecule, they targeted a very specific safe location on the antibody, the Fc glycans.
Fc glycans.
Yeah, these are sugar chains located right at the base of the antibody, far away from the regions that actually interact with the tumor. They used 2 specific enzymes, beta-1704 galactosidase to trim the existing sugars, and Gal-T to attach a new modified sugar containing an aside molecule.
So they basically pruned the sugar tree and put a precise hook on it.
Exactly. And finally, they used a highly controlled chemical reaction called DBCO to attach the click tags exactly onto those asides.
So instead of throwing Velcro randomly and hoping it sticks, they chemically engineered the exact base of every single antibody to hold exactly 2 tags uniformly every single time.
Perfect. Uniform placement. And the biological result was night and day. Because these site-specific antibodies looked uniform and structurally natural, the liver's macrophages just ignored them. Liver accumulation dropped dramatically. The liver enzymes, ALT, AST, and ALP stayed totally in the safe, normal range. Furthermore, the mice showed no weight loss, which is a primary indicator of systemic safety in these models.
That is an absolute masterclass in biochemical engineering.
Yeah.
Okay, final recap for our listeners. Bullet 1, the click therapy successfully reversed aggressive drug resistance in live models by using that Trojan horse method.
Yep.
Bullet 2, upgrading the manufacturing from random lysine attachment to site-specific enzymatic attachment on the Fc glycans saved the liver from toxic accumulation. So audience, here's a thought. Why is the manufacturing of a drug sometimes just as critical to a patient's survival as the underlying science of the drug itself?
It's a great point. And when you synthesize all of this, the bioorthogonal chemistry, the Trojan horse internalization, the site-specific safety profile, you realize we are looking at something much bigger than just a single new drug. We're looking at a modular platform.
But because traditionally, if you want a drug that hits 2 different targets simultaneously, you have to engineer a bispecific antibody.
Yeah.
Right? That takes years of research, massive developmental costs, and you have to start from scratch every time you want a new combination.
Exactly. But with this platform, you don't have to start from scratch. It's Lego-like. You can take existing, highly characterized, FDA-approved drugs, you chemically attach these tiny TCO and tetrazine tags to them, and suddenly you can mix and match them.
That is so cool.
You can pair an EGFR target with the HER2 killer today. Next year, you could swap out the payload, or pair a PD-L1 target with a totally different mechanism. It is an infinitely flexible, cost-effective way to outsmart the chaotic heterogeneity of tumors.
We have covered incredible ground today. We started with the frustrating clinical reality of patchy tumors and receptors that literally barricade themselves. We explored how bioorthogonal click chemistry allows molecules to ignore the human body entirely until they find their matching puzzle piece. We watched molecular Trojan horses piggyback toxic payloads into cells that actively tried to reject them. And finally, we saw how refining the biochemical engineering to be perfectly uniform reversed resistance while keeping the liver entirely safe.
It really represents a paradigm shift in oncology. We are no longer just delivering finished therapies. We are delivering the components and letting the living body act as the final assembly line.
And that leaves us with a truly provocative thought to consider for everyone listening. If we can use bioorthogonal chemistry to turn the human body into a localized test tube, literally assembling cancer therapeutics precisely on top of a tumor, what else could we build inside ourselves?
Oh, the possibilities are endless.
Right. Could we inject dormant, harmless pieces of a vaccine or complex tissue-regenerating compounds? and only have them click to life exactly when and where the body detects an injury. The frontier of medicine isn't just about what drugs we can invent, it's about where we allow them to build themselves. Thank you for taking this deep dive with us today. Until next time, keep questioning the world around you.